Welcome back to another video. So in this video, we're going to discuss about a technique, S1 mapping of a transcript. So to understand what is S1 mapping and what are the different kinds of enzymes which are needed in this procedure, so let's understand this process one by one. Now, first and foremost, what is S1 mapping and why do we need it? Now, whenever you have a transcript and you want to locate the site of initiation, let's say you want to locate 5' end of a transcript, or for that matter, if you want to know the 3' end of a transcript, for that we require a technique that is the S1 mapping. And now this simple pictorial representation, it here shows us the entire procedural which is needed in order to perform the technique. So first and foremost, how it's done now. So step number one, that is, you can see that here we need to generate a probe that will be complementary to the transcript of your interest, from which you want to either locate the 5' end or the 3' end. So in both of these scenarios, whether you want to locate the 5' end or the 3' end, there are minute differences. But here our focus it would be to locate the 5' end of our transcript. So let's start from the first step. Now, in this case, you have a double-stranded DNA, and what we need is we need to synthesize a probe from it. Now we have different restriction enzyme sites, which you can see from here. We have two BAMH1 sites, one over here and another over here, as well as a SAL1 site here. So these are the restriction enzymes. Next is, so in the first step, that is the restriction digestion by restriction enzyme, restriction endonuclease, that is the BAMH1, that is carried out. Now this BAMH1, it will cleave this DNA on these two regions. One is at point here, another is point here. And you can see now this has basically cleaved this double-stranded DNA. Now, next step will be that this double-stranded DNA, it's treated with an enzyme, that is the alkaline phosphatase. Now the functioning of alkaline phosphatase. So this alkaline, now the important thing here is that at both the 5' ends, so if we take this as a double-stranded DNA, so this will be 5' end, and again at 3' end, here it will have the antipolar nature, so this will be a 3' and a 5' end. And both of these 5' ends, these are removed by the alkaline phosphatase. Now, we need to remove it because we use another set of enzyme, because we need another set of enzymes that will incorporate a phosphate, but that will be the labeled phosphate. And that is done by another enzyme, that is polynucleotide kinase. Okay, so polynucleotide kinase, it basically incorporates this labeled phosphates at both the 5' ends. So again, here you can see this orange dot, that is the 5' end, which is now radio labeled, as well as on this region you can see this 5' end that is labeled. The next step is the digestion carried out by another restriction endonuclease, that is the SAL1. Now see, the restriction digestion is carried out and you will get these two fragments separated out. Now, what was the point of this restriction digestion, which was carried by this SAL1? Now, what you can see is that this left fragment, we are not concerned about this, but our main concern is this one, so this right fragment here. Because once we denature this DNA, what we'll get is we'll get our probe, and that probe it will be labeled at its 5' end. So that was our concern. Now, as we have our probe and that probe it's labeled, what we need is the next step should be we get our transcript, the mRNA, and we allow the probe to hybridize to the transcript. So as you can see here, the hybridization will take place between the probe and the transcript because of complementarity. Now again, next thing is, now you can see the actual role of the S1 nuclease, it comes into play at this step. So in both of these cases, so you have an extended region on this right end, you can see here, and this extended region, it's broken down, it's cleaved by a nuclease, S1 nuclease. And similarly, we have this region, this extra region, the tail region of the green end, the probe, that is also cleaved by S1 nuclease. Now that is a differentiating property of S1 nuclease, that whenever it sees a single-stranded DNA maybe, or for that matter a double-stranded DNA or an RNA, it cleaves those. But Only the hybrid formed between the DNA and RNA that is not able to cleave. Now you can see, so these ends here and these ends here, both these ends, these are cleaved, and what we get is we get a hybrid between the DNA and the RNA. Now the next step is this clean fragment. Let's now we take this and we denature it, and this is subjected to electrophoresis. Then afterwards followed by the autoradiography. Now from the electrophoresis, what we'll get is, as per the molecular markers, we'll get a particular size of this separated DNA fragment. And now that fragment, it will tell us where exactly is the location where from the initiation starts of our transcript. Because we know that BAMH1, it has cut at this point, let's suppose. And let's suppose here what we got our result, that was, this is showing that our electrophoresis product that comes at a band of 350 nucleotides, let's suppose. So that means from this BAMH1 site up until this region, we have a 350 nucleotides, and we can easily say that this is the region where the transcription it begins. So this was the entire process and procedure of S1 mapping and why we conduct it. Again, we can perform the S1 mapping in order to locate the 3' end of the transcript as well, but that is with a slight minor difference in between. And we will talk about the S1 mapping of the transcript, but when we have to locate the 3' end in the next video.